Okay, so this lecture is going to basically be on non-ruminant digestion and how they're going to digest protein into amino acids, dipeptides, and tripeptides. And then the next section we'll talk about how they absorb them. Previously, we kind of introduced the idea of amino acids and protein and how they're built. So, digestion. In the non-ruminant, basically has 3 steps. The first step is denaturation. And denaturation is basically going to take place in the stomach, and it's going to be the addition of acid, or HCl, and that is going to expose the peptide bonds. The next step is going to be hydrolysis of peptide bonds. And that is going to occur in the stomach, and also, so the denaturation is all the stomach, and hydrolysis of peptide bonds will be the stomach and the small intestine, and this is done by enzymes. And these enzymes are I'm misspelling it. I'll write it here. So proteolytic, Proteolytic, proteolytic. So these are enzymes that digest protein and break peptide bonds. Okay, and then the third step is absorption of amino acids, dipeptides, now known as di, and tripeptides. And so that is a very good thing. And from there, what we're going to start talking about is what happens in the mouth and get more into specifics. So these are the 3 general big steps, and then we have some specifics. So in the mouth, not much happens. What we are going to have happen is we're going to decrease particle size. We then move to the stomach. And we've kind of talked about the stomach already, in that we have acid release, or HCl, and that denatures protein. And so that's going to disrupt the quaternary, tertiary, and secondary structures of amino acids. We then have pepsinogen, and pepsinogen is an enzyme. It's going to be released. It's actually a zymogen. It will become an enzyme, and it's going to be released by the stomach, secreted by the mammalian stomach. It's going to be activated to pepsin, and it breaks or hydrolyzes peptide bonds. Okay, or breaks the primary structure of proteins. And that's essentially what happens in the stomach. So what we start out with is a protein, and then that protein is going to be broken into polypeptides, and then eventually, so this taking a protein to polypeptides can happen in the stomach or the small eye, and then after we get to polypeptides, this will only happen in the small eye. We can make amino acids, dipeptides, and tripeptides, which are subsequently going to be absorbed. What we're going to do now is build a little table for the enzymes that come from the stomach and that come from the pancreas. So the proteolytic enzymes, we'll give it a name. Proteolytic zymogens / enzymes. Okay, and we're going to have 1, 2, 3, 4, 5, 6 columns. And so we have the site of production. Site of production. We have the zymogen name. We have the activator, or activators, because some of them have two. We have the enzyme. We have the activity site, or site of activity. And then we have, last but not least, the activity. So the first one we already know, and that is produced in the stomach. The zymogen name is pepsinogen. And so that means it's inactive, right? When it has the full name pepsinogen, it's inactive. It's activated by HCl for pepsin. Activation by HCl is reversible, which means it can become inactivated if the pH gets raised. Activation by pepsin is permanent. The active name of the enzyme is pepsin. So this is the actual agent that's going to break peptide bonds. It's active in the stomach, and its activity is it is an endopeptidase. And so peptidase would be an enzyme that breaks peptide bonds. Endo means it's going to attack on the inside of the molecule, or the inside of the polypeptide. Okay, the next three are all going to be produced in the pancreas. And so we have trypsinogen, and trypsinogen is activated by enteropeptidase. Or trypsin. So we'll talk a little bit more about enteropeptidase in a minute, but based on the name entero, that should start to make you think about enterocyte, and so you should be able to figure out that it's produced in the enterocyte, and then trypsin can activate itself. Once it's activated, it can activate other trypsinogen molecules to trypsin. So we have trypsin, site of activity is the small intestine, and it is an endopeptidase. Again, that means it's a peptide bond breaking enzyme that attacks on the inside of the polypeptide. Next we have chymotrypsinogen. That's bad. Chymotrypsinogen, that's an OGEN. Chymotrypsinogen is produced or is activated by trypsin. Produced in the pancreas, activated by trypsin. The active name is chymotrypsin. It's active in the small intestine, and it too is an endopeptidase. Okay, the last one, also produced in the pancreas, is procarboxypeptidase. is activated by trypsin into carboxypeptidase A and B. It's active in the small intestine, and it is an exopeptidase. Okay, and so an exopeptidase means it's going to attack on the outside of the molecule. There's some other enzymogens that are produced, but these are going to be the four that we focus on: pepsinogen, trypsinogen, chymotrypsinogen, and procarboxypeptidase. Okay, these three being active, produced by the pancreas and active in the small intestine, and pepsinogen produced in the stomach and active in the stomach as well. Okay, we mentioned well, I guess I'll define these for you guys. We have an endopeptidase, which hydrolyzes peptide bonds on the inside of a molecule, on the inside of a polypeptide. And we have exopeptidase, which hydrolyzes peptide bonds on the outside or the end of a polypeptide. Okay, and so that provides definitions for you guys on endo and exopeptidase. The guy in the next office just left, so that's door slamming. Okay, we said we'd mention enteropeptidase. Enteropeptidase has another name, and that is enterokinase. So if you ever see that, you know what that means. It is produced by enterocytes. And so our good friend the enterocytes, and it's produced in response to CCK and secretin. Okay, and so enteropeptidase is what is going to activate trypsinogen to trypsin, and then that releases a cascade where chymotrypsinogen and procarboxypeptidase can be then activated by trypsinogen. We're going to draw a little picture, kind of, or a diagram, a cartoon. So our good friend the enterocyte, so we'll put an E here, just so I remember who that is. It's an enterocyte. Draw a new type of cell in the small intestine, an I cell. Another enterocyte. Another enterocyte. Gonna sneak another cell type in here, and that will be the S cells. So we have two new cell types, the I cells and the S cells. And we'll make one nice enterocyte here, a happy little enterocyte to make the Bob Ross fans happy. And so, as you guys can tell from the microvilli, this would be the luminal side. And so what we'll do is we'll put some digesta on the lumen, label it as lumen of the small intestine. And so we'll put some digesta in here. And so what we want to think about when it comes to protein digestion is put some happy little digesta in our small intestine. Okay. What we want to think about is, what is this digesta? So if we're in the proximal duodenum, what are some of the characteristics of this digesta? So this digesta has a low pH. And the reason it has low pH is because it's leaving the stomach. Okay, and so the pH is low. It's showing up at the proximal duodenum. The buffer from the pancreas and the small intestine hasn't had an opportunity to act yet, because we're actually going to send the signal now to cause those to be released. And the other thing it's going to contain is denatured polypeptides. Okay, so we have acidic digesta with denatured peptides or polypeptides, and what that's going to do is the I cells and the S cells are going to recognize the presence of this low pH digesta with these polypeptides, and the I cells are going to release CCK, cholecystokinin. The S cells are going to release secretin. Okay, both CCK and secretin are going to act on the enterocytes. Okay, and in response to this, the enterocytes will release enteropeptidase. Okay, and so now we have the small intestine releasing enteropeptidase in response to the presence of a protein-containing meal that the animal consumed. The CCK and the secretin aren't only going to act on the enterocyte, but they're also going to act on the pancreas. In particular, the exocrine pancreas, to cause the release of the zymogens and buffer. So the pancreas is going to release zymogens. Oh, sorry guys. Pancreas is going to release zymogens and buffer. Okay, and the zymogens we just said are trypsinogen, chymotrypsinogen, and procarboxypeptidase. And now we have those zymogens present. We're going to raise the pH. So it's going to increase the pH, and the enteropeptidase will activate the zymogens. We'll add a few words about that. CCK and secretin cause enterocytes to release enteropeptidase and cause the pancreas to release zymogens, or 3 zymogens from the pancreas, and buffer. And so the buffer is going to raise the pH, allow the enzymes to be active, and the enteropeptidase will activate the zymogens into active enzymes. Okay, so that's a good deal. One thing we want to know is, if I can fit it, I'll try and fit it on here. What causes a decrease in zymogen secretion? And the answer to that is excess free trypsin. So what free trypsin is basically trypsin that's not attached to a protein molecule. It's not digesting anything. It means there's nothing there to digest. And so that sends the signal to the small intestine, which sends the signal back to the pancreas saying, hey, we have enough zymogens. We don't need to produce anymore to digest any more protein because we've digested it all. The meal's ended. We've digested the protein. Stop producing trypsinogen or sending trypsinogen to the small intestine. The reason we want to do that is trypsinogen is made of amino acids and is a protein, and so are the other zymogens, and so they're relatively expensive to make, and if there's nothing there to digest, you want to stop making them. And so what this is known as is negative feedback. Okay, so it's like a negative feedback loop. It's not like it is. Okay, start on another sheet of paper. Scooch you down here. Okay, and so what we want to do is introduce the idea of trypsin inhibitors. And so trypsin inhibitors are basically going to stop trypsin from being active. Legumes generally contain them, and other feeds contain them as well, but legumes contain quite a bit of them. Where we can get them commonly, no one feeds them because we know this is a problem, but is in raw soybeans. So if you feed, or can say you consume some raw soybeans or a lot of raw soybeans, you will inhibit the activity of trypsin. And so the result of inhibiting the activity of protein or of trypsin is the zymogens are not activated. So your chymotrypsinogen and your procarboxypeptidase can't be activated into active peptidases or proteolytic enzymes. So they're basically just not doing anything. They're sitting there as zymogens, not actively digesting. And so what that results in is in a decrease in protein digestion. So protein digestion is going to be reduced. Okay? The other thing that happens is we don't have a negative feedback because there's no excess free trypsin, because all the trypsin is bound up to these inhibitors. So there is no negative feedback, or there's no excess trypsin, and so no negative feedback from free trypsin. And so as a result of that, the pancreas continues to produce zymogens. Okay, and there's two reasons that's concerning. Well, the first reason, or there's more than two, but one reason is we're not digesting protein, so that's concerning. The other thing that is concerning is synthesis of zymogens requires energy. So we're using ATP to synthesize these molecules, and that represents a waste if they're not going to be used to digest protein. B, zymogens are proteins, and as proteins, they require amino acids to be synthesized. And what ultimately happens is you end up with pancreatic fatigue. because you're using all of your energy and all of your protein or amino acids to synthesize these zymogens that aren't digesting protein, and so you can really compromise the animal's performance. Now, in most practical feeding situations, this isn't a concern because we don't actually feed raw soybeans to animals. We know that they contain trypsin inhibitors, so we process the soybeans before we feed them, but sometimes we'll run into novel feed ingredients or legumes that they might graze and they might consume some of the seeds, and you can have problems with trypsin inhibitors. Trypsin inhibitors are also a bigger deal in non-ruminants than ruminants, because in the rumen, the microbes can deal with, to some extent, the trypsin inhibitors. Okay, so we really like to make sure to not have any trypsin inhibitors because it's going to decrease protein digestion. And if you have really high levels, you're going to have pancreatic fatigue, basically because you're using a lot of energy and amino acids to synthesize zymogens that aren't doing anything. Okay, we have another group of enzymes, and these enzymes are brushy border peptidases, or brush border peptidase. And they're going to be produced by enterocytes. We have 3 of them. We have aminopeptidase, we have dipeptidyl amino peptidase, and then we have tripeptidase. And these are produced by the enterocytes and released into the lumen. And these enzymes, coupled with the other 4 enzymes that we mentioned previously, result in the end products. So the end products of digestion are amino acids, dipeptides, and tripeptides. And so, I need to cross my T. And so what we're going to do on the next presentation or the next lecture is look at how these 3 products, amino acids, dipeptides, and tripeptides, how they're absorbed and what that looks like. So hope you guys have a good day. I hope you guys are enjoying this modified semester, and make sure if you have questions to post them somewhere in the comments on YouTube, or group me would be ideal, or you can shoot me an email as well. And other than that, take care.